Colton is a familiar face around RIEL. He has been here since the end of 2021 and has really become an important feature of RIEL. Everybody knows Colton. And Colton has submitted his PhD just at the start of this year. So let's congratulate Colton. And he has said that now he's got so much free time, he wanted to do a seminar. So he is going to be presenting his PhD research today. So without further ado, I'll hand over to Colton. Thanks, Colton.
Thanks, Steve. Thanks, everybody. Okay. So I'll just blame Julia if she's watching. She presented her PhD and it seemed like a good idea at the time, so I signed up for this. Yeah, lots of free time. I got it eaten really quickly. So today I'm going to be talking about trophic dynamics and free-flowing rivers in the tropics. It was part of my PhD. Keller was primary supervisor, and it was also supervised by Arsmar and Daniel Stratford from CSIRO. Just like to acknowledge the fact that we are gathering on unceded lands and the sovereignty of the traditional owners of these lands, past, present, and emerging. So to start out with the fun news, global rivers and wetlands are critically threatened, and over a quarter of the freshwater fauna is threatened with extinction currently. So freshwater research is kind of an important thing at the moment to try to stem these losses that we're having. What's going on? Oh, all right. And rivers and wetlands in the tropics are under increasing pressure for development. Historically, a lot of the development has been in the temperate north, and now it's creeping into the tropical south and other regions. Whereas tropical Australia is very lucky in having a large number of free-flowing rivers remaining in the tropics. They've not been modified or formed. And this is important because our Australian fauna is not critically threatened yet. Less than 5% of it is, so it's very good. These natural rivers are very good places to provide ideal environments to study how ecosystems function under natural flow, which is a main regulator of community structure in rivers. Flow in rivers is correlated with fish species richness, kind of like your terrestrial species area. Flow acts as a similar mechanism and can be used to look at relationships between area, species richness, and other means of community structure. Also, the rhythmic nature of flooding in these rivers influences food web dynamics and how communities are structured. So based on theory of flood pulse and river continuum and stuff, and those early theories, we created a conceptual model to sort of look at this thing blocking my notes. All right. Cannot move it. There it is. Okay. So floodplains under natural flow are really dynamic ecosystems where you have wet season inundation of terrestrial habitats, which introduces a whole suite of new types of food from the terrestrial environment. We call that allochthonous inputs or subsidies. The inundated areas create migration pathways for fish to utilize for reproduction, feeding, other things. You have longitudinal connectivity where the floodwaters coming down from the headwaters are bringing with it materials that provide energy for primary production. And another important aspect of this is you have groundwater recharge, you have a reduction in saltwater intrusion, so you have these expansive salt pans that become fresh and produce a new source of food input during periods of inundation. What happens when you develop these ecosystems is you lose a lot of that natural function. So you have reduced conductivity, reduced inundation area and inundation times, an associated increase in turbidity because there's a change in land use. Another impact is that there's an associated land clearing so that those terrestrial energy sources and food sources that were inundated and contributing to the aquatic ecosystems may be removed. You have less habitat for insect fall, less fruit fall. So there's just overall a lower input of food sources and growing area for in-stream autochthonous food sources in the form of algae. And you also have added negative impact of increased saltwater intrusion, which changes those freshwater wetlands in the terminal end of these river systems. Then in the dry season, it's a completely different situation. Their base flows are maintained by those groundwater supplies that were topped up during the flooding. So really important, that groundwater connection, and that continues to supply longitudinal supply of materials from upstream, but also it maintains wetland habitats that are no longer connected to the main river. And you have a general shift from food coming from the terrestrial environment to food being produced in the aquatic environment. And these wetlands that are supported by groundwater are also important refuges during these dry periods. Also in the bottom end, you have this transition back into a brackish or estuarine environment, which brings in a whole new suite of fish and food sources. Again, looking at that, when you modify and do through water extraction, you're reducing the permanence of these wetlands, which can cause fish kills. You're reducing the availability of habitat to provide inputs through terrestrial sources like insect fall and fruit and leaf litter, because you've largely cleared that. Generally, if you go in for water extraction, there's that associated land use changes. There's an increase in potential for fish kills and a further increase of saltwater ingress up the main channel. So understanding trophic dynamics in these aquatic ecosystems is really important as pressure increases in the tropics to develop these systems. So if we can get a baseline handle on how these ecosystems function, we can hopefully manage them. So the aims of this project were to examine river discharge, which is known to influence fish species richness. And I examined how multiple discharge metrics influence species richness across different trophic levels of the fish community. I also wanted to compare isotopic trophic niches of fish communities in the Roper River. So looking at how the niche area used by river fish and wetland fish communities changed over different flood magnitudes and between river and wetland habitats. And I wanted to compare food quality, which is assessed through fatty acid analysis, and how food quality is changed through the influence of flooding of different magnitudes and between the river and wetland habitats. So to examine tropical rivers are under increased pressure, as I was saying, for development, and these highly biodiverse ecosystems and understanding how river discharge influences these fish communities is important and useful in managing systems as we go forward in time and increased pressure to develop. So I've published this paper on how species richness of each trophic guild changes or is influenced by different flow metrics of the river. And I did this by looking at the trophic theory of island biogeography, which is looking at the area species area relationships. And it postulates that the slope of the species area relationship should increase for predators, because predators rely on densities of prey and tend to be rarer than others. So they're much more sensitive to changes in prey density and environmental factors. Few studies have tested if the species area relationships vary across trophic gills. And here I attempted to do that across tropical northern Australian catchments. So the study area that we did, we have data, I collected data from previously existing work by Sternberg and Kennard, and updated the taxonomy and some other flow metrics. And we looked at 40 catchments. We had flow metrics for 40 catchments across the 3 drainage basins of the tropical regions of Australia, northern Australia. Then we modeled those river discharge metrics for the mean annual discharge, the mean wet season discharge, mean dry season discharge, and the coefficient of variation of the mean annual discharge, which is a measure of variability over the year. What we found was a total of 127 species from 27 families. Not surprisingly, herbivores and invertivores and omnivores made up the bulk of the species richness, 72%, whereas herbivores and predators accounted for 10 and 18% respectively. And so this is kind of an indication of where we're going, that these rivers, even though they are unmodified, Australia is characterized by highly variable environments, and so dominated by resource generalists rather than specialists. We analyzed this data using a generalized and linear mixed model, looking at species richness as a function of trophic gill, flow metric, and drainage division. So drainage division was added as a random effect to sort of account for those random. Sorry, that is not my language. We'll keep going. So the results, what we did find is that mean annual discharge was significantly correlated with predators, invertivores, and herbivore detritivores, however, not for omnivores, which I guess is kind of expected. Omnivores are a generalist. Then if you go down and you look at the dry season and the coefficient of variation or variability in flow, only invertivores and omnivores were significant for those variables. So they're interacting with that variability, whereas the other 2 gills were not. If you look at these fitted linear models, and you can see the slope. So this is for wet season discharge, which in our tropical environments is mostly considered or very similar to the mean annual discharge. So I'm just presenting the wet as sort of mean annual discharge. There's not a lot of variation in slopes, which is what we were looking for, and you can see that the omnivore kind of flattens out for the wet season or the high flow periods, and that fits with their ability to be adapted to variable environments. Then when you look at the dry season, the slope for invertivores and omnivores increases a bit, whereas the predator in the herbivore detritivores sort of flattens a bit. And that's just again showing how these groups of animals are adapted more towards that variable environment. And again, we had an opposite effect for the increased variability in annual flow, which is what you would expect. So increased variability has a negative correlation with species richness across the trophic gills. But again, each one of these, we didn't have a huge variation or a significant variation in the slope curves for any of the trophic gills. So in summary, the results show that species richness of trophic gills in the North Australian freshwater fishes did correlate with multiple components of the wet and dry tropical river discharge. However, predators did not have a significantly higher slope, and rather the gill slopes tended to be similar across flow metrics. The observed increase in predator species richness in wet season discharge highlights the importance of flooding for this gill, specifically for feeding and recruitment, whereas omnivores were more abundant and likely better suited in Australia's unpredictable and variable environments during the dry and under dry conditions and variable conditions. And this shows that tropical river discharge in Australia is highly variable, and predictions of the trophic theory of island biogeography may have limited applicability in these environments because we're dominated by resource generalists rather than resource specialists, so the reaction is less pronounced. Overall, the results support the hypothesis, suggesting that lower river discharge and higher annual flow variability play key roles in explaining species richness, as Australia is characterized by having low species richness in tropical environments compared to other tropical environments, which reflects our flow variability, but also reflects other, biogeographic and phylogenetic aspects, which I won't present here. And these results suggest that maintaining dry season flow and managing increases in flow variability will be an important aspect of managing and conserving trophic species richness diversity across tropical Australia. And I say that because, with invertebrates and omnivores, that 72% of your species richness are significantly correlated with those flow variables, and therefore they're quite important to manage. Moving into more specific questions about food webs, I did some very specific work in the Roper River and investigated the influence of flood magnitude in river and wetland habitats on fish community food webs using stable isotopes. So this is important because connectivity across the river floodplain matrix is known to support very high biodiversity and productivity. And we know that the annual flood pulses are important catalysts for subsidies into the aquatic food webs. And that annual connectivity may influence fish community food webs through shared resources. So that connectivity and that abundance of inputs should have some effect on how the community trophic niche is shaped. So just a little bit of background on the theory of these things. The flood pulse concept and the river continuum concept were early ideas and based on the flow of energy. They emphasized the subsidy of allochthonous materials coming in. The perception was there is an abundance of those materials coming in through flow from upstream areas, and then that must be important as a food source for the aquatic environment. However, more recently, looking at trace elements, it's become quite apparent that algae is quite an important contribution, and that algae is a autochthonous produced within the aquatic environment, and it has a great influence on the productivity and biodiversity of aquatic ecosystems. When you put this together in a unifying theory like the river wave theory, you can see that it's actually a combination of terrestrial inputs, autochthonous inputs across the variation of the hydrograph. So as the hydrograph increases, you would expect that there's going to be a heavier influence of allochthonous inputs coming from the terrestrial environment. And then as the hydrograph recedes, your food web becomes more dependent on your internal or autochthonous production of algae. So using the stable isotopes helps us understand the flow of energy and how that structures aquatic communities. Stable isotopes of carbon are used to give an estimate of the source of the food Enriched carbon indicates an aquatic or autochthonous food source, and less enriched a carbon is more indicative of terrestrial or allochthonous inputs. And then your nitrogen can be used to estimate trophic position, or we use nitrogen to estimate trophic position. So you create these polygons in to compare how different communities are similar or different based on multiple different aspects of inputs of carbon, sources of carbon, diversity of carbon, and trophic position, which is measured by the total nitrogen. That one, sorry. So to put that sort of those theories that I just talked about flood pulse and river continuum and the river wave, created a bit of a conceptual model on what you would expect during low flow and high flow conditions in wetland and river habitats. So in low flow, it is expected that you would have a narrower carbon range, lower trophic position, feeding at lower levels of nitrogen. The community would be feeding on more in-stream autochthonous sources of food, and there would be less overlap between the river and the wetland habitats, obviously because they're isolated, and they're not likely to be sharing any food resources. Then under high flood wet season conditions, it was expected that the trophic niche of the communities would expand, and there would be a broader base of carbon. The consumers would be feeding at a higher trophic level, and there would be more overlap between the river and wetland fish communities because they're sharing a common abundant food source. So I did a study 4 wetlands and 2 river sites in the Roper. I did some opportunistic studying at the runoff period in 2023 at a drain in the river and a drain outside of one of the wetlands, Walmaja. And so that, just to give you an idea, the Roper River is southeast of Darwin. It's a Gulf drainage, and it's one of the largest remaining free-flowing rivers that is perennial in flow. It's got a massive amount of groundwater flow, so it's quite an important river. It was really interesting to look at. And so for this chapter, the stable isotopes and the fatty acids, these sites are all the same. The fish were the same. I collected fin clips for isotopes, and I collected tissue cores for fatty acid analysis. I was really lucky in my years because I wanted to look at effects of different flow. So in 2022, the flood magnitude was about 33% below average, whereas in 2023, we had a flood magnitude peak discharge 82% above average. So it provided a really good comparison. Here you have one of the main lagoons, Lamarium, which is a really beautiful long lily line lagoon. You can see in 2022, it got quite low. This is June 2022. This is June 2023. The water level was quite high. So really good comparisons. Walmaja, which is another very interesting lagoon, dried up completely at the end of 2021 or 2022. And then in 2023, it was still full, and we got a decent sample out of it. So it was I was very lucky in the comparisons for flood magnitude. Uh-oh.
Screen.
Yeah, we'll figure it out. Thank you. Okay. Yep. Okay. So we estimate the trophic position for each individual fish based on basal food samples collected at each site. and then you calculate a trophic position for the fish, and then along with the carbon, you can create these Bayesian ellipses to look at the shape, the breadth, and other measurements of the trophic niche. And then to further analyze how those things changes, you use layman metrics, which are more detailed examination of the measurements of the niche area, such as niche breadth, which is a measure of the whole food web, carbon range, which is how wide they're feeding across different food sources, and a measure of redundancy in packing, so you can get estimates of the diversity of food the community is consuming. So to get the results, I collected 680 individuals from 26 different species and 16 families. So it's a pretty substantial community database, and it was a good opportunity for us to look at a large number of fish across communities, and those communities were broken into river and wetland and high flood and low flood conditions, because a lot of isotopic research has traditionally been based on lower numbers. So we tried to get as many as we could across as wide a range of size classes and other things. Here we're just presenting the community stuff, but there is likely going to be more stuff to be able to come out of this because we did collect so much data. So creating the community polygons, really interesting result. The river at low flood, which is this big one here, actually expanded. It had a very wide carbon range, which is on the x-axis, and the y-axis is the trophic level. So during low flow, the river the community was feeding at a higher trophic level across a broader range of carbon sources and a broader diversity of carbon sources. Then the river at low flood, there was a contraction. So a narrowing of the carbon range and the total breadth of the niche area, and a lowering of the feeding, the trophic position at which they were feeding, the community was feeding. And as kind of was expected, there was less variability across flood conditions for the wetland, and the wetland trophic area, niche area, is a bit more similar to the river at low flood, or at high flood. So that indicates a sharing of abundant resources. And then further analysis with the layman metrics just show what specifically was driving these three specific areas that were driving this. So the total area was significantly higher for river at low flood. Total carbon range was very, very significantly different. So the river is feeding at a much wider carbon range, and the diversity of things consumed, the trophic diversity, was significantly different for that river at low, whereas the other ones are all a bit more similar and not so significantly different. So something significant is going on in the river at low flood. So in summary, the flood magnitude did substantially influence fish communities' isotopic niches, especially for the river at low flood. Surprisingly, contrary to what the idea and the conceptual model is that the river at low flood expanded its trophic niche, breadth, its carbon range, its trophic position in which things were feeding, and the diversity of things that were being fed on. And this may be the result of an increased production in algal in the absence of flooding. Flooding has a tendency to rip out those, to disturb and remove algal food sources. And then you have an input of terrestrial, an abundance of terrestrial or other forms of algae during flooding. And then during high periods, what we suspect is happening is that because of all the inputs of the flood material, that the community, both river and wetland, are sharing those, so you get stronger overlap. So yeah, it's an interesting variation, and kind of it was a bit unexpected to see that broadening of the niche in the river at low flood. Well, in conclusion, for these niche analysis, both lateral and longitudinal connectivity play a key role in subsidizing food webs in the Roper River. So both flooding and non-flooding environments provide a different source of food for the community. And that low magnitude flooding allows these algal communities to increase in their contribution to the food webs and expands the trophic niche, while flooding narrows it. And it's that indication of just an abundant shared, less diverse food resource during flooding. And it demonstrates the importance of understanding how floodplain river food web dynamics under natural flow. If we can understand these, we can try to incorporate the importance of these connectivity issues and flow issues in maintaining river biodiversity and food webs as we develop these rivers, because it is happening. So a bit more detail was then sought to look at how food quality and more specifically food source was influenced by flooding and habitat types, but using a different approach, which is fatty acid analysis, which is a little bit more refined in determining where the food's coming from. And certainly because of the nature of the fatty acid, it indicates quality of the food. And flooding influences the essential fatty acid. The reason why we call it fatty acids are useful in determining quality is because fatty acids are metabolically necessary for fish. Therefore, they're essential, and they can be used to determine food quality. And understanding how food quality in natural flowing rivers is vital in managing aquatic ecosystems as they are being developed, because those developments, going back to that conceptual model, if you're reducing inundation times, connectivity, and water quality, you're going to have an impact on food quality, which will have an impact on the community, food web, and health. So while isotopes are used to detect carbon sources, trophic position, quality of food can be more, as I said before. So certain fatty acids are metabolically necessary and difficult to synthesize, but much easier to consume, such as EPA and DHA. There are two specific fatty acids, ALA and LA, which cannot be synthesized by heterotrophs and must be consumed. And they're also really important because they're precursors to EPA and DHA, which are metabolically required for reproduction, neurological development, somatic growth, and they're very important. So these proportions are really useful in determining how healthy a food web is and what the quality of the food coming in is. You can also look at total omega-3s, which you guys have all heard of, and omega-6s. Omega-3s tend to indicate aquatic food resources, and omega-6s indicate terrestrial food sources. So I looked at those as well in my models to determine where food was coming from and an estimate of quality. So using the foundational theories presented in the previous chapter, I developed a hypothesis to test here. Suspected that food quality would decrease with flood magnitude as measured by the proportion of these fatty acids, especially long-chain polyunsaturated fatty acids, which are EPA, DHA, and ARA. And that would be different in river and wetland habitats. And wetland food webs would have a lower quality as determined by long-chain polyunsaturated fatty acid content when compared to the river channel. So again, a large database. The way that we approached this was to analyze the whole of the food web. So we had 116 primary producers and 150 primary consumers or invertebrate consumers across 28 species of fish from 17 families. A total of 412 fish tissue samples were analyzed in the food web. And that again was broken into river, wetland, high flood, low flood. We were and just analyzing the essential fatty acids. So there was 5 essential fatty acids: EPA, DHA, ARA, ALA, and LA, and then total omega-3 and total omega-6s. That was modeled using a generalized linear mix model, and we looked at the function of habitat type, taxa, and flood year, and used site and month as randomized effects. The model summary, what we found is that ALA, ARA, and DHA were significant for flood magnitude, so they tend to change according to flood year, large floods, low floods. And ALA, ARA, and total omega-6s were more were significant for habitat. While not significant, and considering the size and variability of this data set, all other fatty acids showed similar trends for river floodplain habitat, suggesting habitat is a major driver of food quality on these ecosystems. So more specific, sorry about the image, I could not get it to work well. So what this is looking at is flood magnitude, and this is DHA and ARA, which are both long-chain polyunsaturated fatty acids, which are useful in determining food quality. And across the whole of the food web, it shows that food quality is improved because of the proportion of these things in low flood conditions. And so that's quite significant. And again, for habitat types, we showed that so your habitat, river, and wetland, and then total omega-6s, which is an indication of terrestrial inputs, and ALA, which is an indication of terrestrial inputs. It's accumulated in invertebrate consumers and then consumed by fish. Those both were consistently higher in wetland habitats, so suggesting wetland habitats are driven more by terrestrial inputs across the whole of the food web, considering all averaged across all those animals and all the other variables. So it's kind of a cool result with the food web. So the takeaway are that wetlands and high flood magnitudes provide poorer quality food while maintaining metabolic necessary essential fatty acids, because all of the food web still had those. This challenges the idea that algae are the only important food source, and that ALA and is important because ALA and LA, which are used to synthesize those longer chain polyunsaturated fatty acids, are being sourced when those high quality algal sources are absent. Therefore, it's suggested that the quality definitions for tropical ecosystems should be re-examined. And this is based on the fact that most of our understanding of food quality comes from temperate regions. Hydrological connectivity maintains diverse energy pathways. High magnitude floods equals allochthonous subsidies, so quantity over quality. And again, providing those metabolic pathways to create the needed long-chain polyunsaturated fatty acids for those metabolic functions, even if they're not in the food source. Low magnitude floods, you have autochthonous inputs, so quantity over quality, quality over quantity, and then both pathways appear to be necessary to create food web resilience in these ecosystems. Management implications that natural flow variability and floodplain connectivity are critical. Lateral and longitudinal connectivity support diverse food sources, and healthy riparian and wetland habitats enable those metabolic conversion of essential fatty acids to the long chain polyunsaturated fatty acids when algal food sources are disturbed by high floods. Flow regulation may reduce food web resilience. Therefore, maintaining some flow variability and connectivity to the wetlands will be important. So generally speaking, we know that globally rivers are under threat and are losing biodiversity more rapidly than any other ecosystems. The rivers of tropical Australia have provided an ideal study site for how aquatic trophic dynamics are influenced under natural flow discharge. The trophic ecology and its relationship to river discharge underscores much of the ecological function in large floodplain river ecosystems, including mapping the flow of energy through the food web and how that relates to the consumer community. This thesis examined fundamental theories of trophic dynamics and freshwater fish community structure in relation to natural flow and river discharge. Chapter 2 highlighted how flow influences trophic species richness, especially during the dry season, and negatively by the increased variation and annual discharge. Chapter 3 revealed the importance of hydrological connectivity and maintaining diverse energy pathways, supporting these fish communities across the river floodplain habitat complex. Chapter 4 found that tropical floodplain river food webs rely on both high-quality algal autochthonous inputs as well as lower quality but far more abundant allochthonous subsidies during high floods. Understanding how trophic dynamics are influenced by river discharge and annual flood cycles can be used to manage and conserve floodplain river ecosystems under increased development pressures, as we're seeing now. Maintenance of dry season flows while reducing variation in discharge will maintain trophic species richness. Lateral connectivity provides an abundant shared resource for consumers, river food webs, and become more diverse when less disturbed by larger floods, whereas water resource development should include diverse discharge levels and regular connectivity to wetlands when modeling hydrology for extraction so that you can maintain those connectivity and diverse flow rate of discharge regimes. Food quality will increase with lower disturbance from high flood magnitudes. However, inundation of wetlands and the abundant allochthonous subsidies also support food webs during flooding. Therefore, maintaining both lateral connectivity, flood variability, and wetland habitat quality must be included in management of the resources. Future needs, things that would be good to do would be incorporate fish trophic species richness data and added life history data from other tropical regions and associated hydrological metrics to validate the trends observed in Australia. May or may not happen. Australia is lucky in that we have access to all these unregulated rivers. It's relatively good, and we also log the hydrological metrics. We also have a pretty substantial understanding of taxonomy and distribution of our fish here. That's not necessarily present in other tropical regions as of yet, but it would be good to compare. We can improve the understanding of community trophic niches by increasing basal food resource samples, especially benthic algae, and increased temporal sampling because there's a lag in the tissue uptake of these things, and they change over. So more regular sampling, especially during the wet season, will refine the understanding of how the food production and community are consuming resources during flooding. And then food quality conditions should be broadened for the tropics, as we are high flow, high turbidity ecosystems here in Australia, and a further understanding of the metabolic processing of essential fatty acids is coming largely through the aquaculture industry. And this will give us a more detailed understanding of the retention and use of low proportion of essential fatty acids found in tropical river floodplain ecosystems, because those essential fatty acids do tend to be lower in proportion in the food sources of the tropics. In conclusion, free-flowing rivers have evolved dynamic relationships between annual flow variability, habitat diversity, and that maintains a high biodiversity within aquatic ecosystems. Flow and source of energy that support high diversity of fish and other biota are influenced by the annual flood cycles and diversity in floodplain habitats that subsidizes the aquatic ecosystem. When any modifications are planned for these free-flowing rivers, the natural variation in flow, which have created a balance in the aquatic food web, must be considered if these rivers are to maintain and support both human and ecosystem needs. Thank you. I would like to know.
I definitely learned lots of new words. And I think you're probably one of the few people that's benefited from the weather during your PhD.
Well, it was planned. Keller put out an order.
A really dry wet season and then a really wet season.
It was lucky.
He also was in the field on the hottest, in the place one year that was the hottest place on the planet that day.
Nooka was in October 2022. Nooka, Keller told me later that Nooka was the hottest place on the face of the planet. So I had Jimmy, who's a mountain of a man, and Tess Pointer, which some of you may know, up the front of the boat, a generator in the middle, an outboard behind me, full sun, working 10 hours in the sun. There was not a dry spot of clothing on any of us. It was pretty brutal.
Thanks, Colton. So we're going to have some questions now. If you are online, I will get to your questions after we answer some questions in the room. So feel free to type your questions up now. Start with Sunil.
Thanks, Colton. That was great. I was wondering, given the variation in food quality associated with season, and I was wondering whether, you've classed all of these fish species into different trophic levels. And of course, you have a size range, so you know that. But I wondered whether in reality, some of these species, or maybe many of them, actually change their trophic level with life history stage.
Absolutely. There's quite a few papers coming out now. One specifically I can think of is bony brim, which is a really abundant herbivore, and they shift trophic position across their life history. So in early life history, they're feeding on zooplankton. Then they'll go into a herbivorous dominant feeding pattern, and then they'll go back to another more predatory stage at a large. I think that's how it goes. And so that was one of the intentions of collecting across the size class so that we could break that down. What I presented here is a general food web. And the other thing I think that's really important is in the tropics, I think there's a different set of fatty acids that may be a better gauge of food quality, because in a turbid, predator-rich environment, growing quickly to become the predator, to shift into that ontogenic stage where you're eating other things is more important. And that is driven, as you might know, in aquaculture by your saturated fatty acids. So, the fish are always going to be able to, at some extent, especially in the tropics, in freshwater, synthesize the long chain things that they need when they need to reproduce, when they need to have to develop their neurological systems or whatever. A lot of that neurological stuff from EPA, DHA is based on temperate systems that are clear water, large-eyed fish that are feeding on very small prey. We are in a turbid environment with bucket mouths sucking in as much things as they can in an environment which they can't see. So it will be interesting as these ideas develop and when we start stepping away from this concept of algae as driving primary productivity and biomass in tropical environments, because here we don't have those slow, long-lasting, inundated areas where a whole bunch of algae can grow, where you're having a massive amount of fruit falling in, specialists that eat it. There's so many different things happening here that there might be a different suite of things to look at. And I do think from the results there that terrestrial inputs are quite important, and they are sources for those long-chain PUFAs, but through metabolic conversion rather than consumption. Sorry, that was a long-winded answer.
Thank you.
Oh, that's fantastic, Colton, and it's wonderful that you've produced so much information on a remote area, on a river that hasn't been studied, because so little of that work's been done in the north, particularly in the NT. My question, sorry, just quickly is, and I just want to continue that theme of turbidity, because clearly from all your photos and everything that most people here who go out into floodplain rivers know, is that the flow is highly correlated with turbidity. And that turbidity is delivering nutrients to the floodplain, but it's also limiting algal growth because it's huge light limitation. So it does need and because they're highly correlated, it's going to be really hard to separate the impacts of light limitation from flow.
And I think,
So how would you do it? Or that's a question for a whole nother PhD.
I mean, that is a question for, and one of the things that I didn't say it, but it was in the future sort of directions, would be to look at compound specific amino acid analysis, where you can actually get a much clearer answer of where that food really coming from a terrestrial source, or is the theory that largely the animals are just acquiring algal food sources, because a lot of the invertebrates are concentrating algal food fatty acids and then being consumed. So yeah, it is possible, but the reason why we don't tend to do compound specific amino acid analysis is because it's very expensive. These fatty acid analysis sit at about, I think, $50 a sample. Compound specific, I think you're looking at 192. So you're not going to get the same numbers and same resolution, but to be able to do that, I think is really important, because the other thing that I think is not talked about that much is the algal food sources up here are very, very low in proportion of those long chain polyunsaturated fatty acids. But they're, I didn't show it, but the proportion of fatty acids in my fish were over 50% saturated. So SAFAs, the lower number, which is not deemed as significant or essential or quality. But the proportion of those in the fish tissue was very high, higher than tropical reef fish, which has traditionally been seen as the highest SAFA. So there's a question there. And I think that is because of that turbidity, that a lot of that terrestrial input is actually fueling the floodplains up here.
Any other questions in the room?
Well, thanks, Colton. You mentioned really briefly that there was a time lag between the time a fish eats a food and that shows up in the tissue. So how do you know what you're sampling? What time of
So based on a lot of the literature, we sort of have a time frame for carbon and nitrogen in tissue. And so that June sample, we would expect that we were looking at sort of the end of the dry season food consumption, or the end of the wet season food consumption. Then later in the year, you're looking at what the dry season food would be. So I think with carbon and nitrogen, you're looking at weeks to months. With fatty acids, you're looking at months, a little bit longer of a turnover rate. And that's and the issue with that is that these tropical environments, currently we can see it's really hard to get out on a river in the wet season to see what's being consumed at that specific time in the wet season. And that's something that we tried to do with track and nest by having motherships and going out onto the floodplain with smaller boats to try to do that. But there's not many projects that have that kind of money to be able to do that, because most of us are bound by land to get to those sites to collect that data. And that's what I was suggesting, is more temporal sampling to try to actually get better resolution on how that uptake changes over the hydrograph. Because right now we're just guessing based on dry season sampling.
And we got some online questions. Anybody else in the room? Okay, and so I would like everybody to join me again in thanking Colton. Let's have a little token of appreciation. And I would also like to remind everybody that morning tea is on this morning, and our seminar series will be continuing next fortnight. So keep an eye out for the next presenter. Thank you. you.
Thanks, everybody.
I know, but you're going to be traveling. I didn't know I'd see you at work.
Ever. So that was
Nice surprise. Well,